For nearly 2 decades, Asian carp seemed unstoppable. They overwhelmed American rivers and rewrote entire ecosystems. In some stretches of the Illinois River, they made up most of the fish biomass, and every survey showed the same relentless rise. Every barrier failed. Every harvest program fell short. Every expert said the same thing. This invasion had no ceiling. Scientists tracked it. Managers fought it. Politicians funded programs against it. Season after season, the numbers climbed. Survey after survey confirmed the same trajectory. In some stretches of American rivers, these fish had stopped being an invader and started being the ecosystem. Then something changed. Not dramatically, not everywhere, but consistently, in specific corridors, the metric that had only ever moved in one direction started moving the other way. Nobody had ordered it. Nobody had planned it. No new barrier had gone up. No budget increase had funded a larger harvest operation. When researchers finally traced back what had actually happened, the answer led to a decision made in 2004 that had absolutely nothing to do with Asian carp. A program built around an entirely different goal had been quietly running a carp suppression operation the entire time. It just didn't know it. My name is Jonas Flint. I have spent 31 years walking river systems in this country, from the backwater sloughs of the lower Mississippi to the spring-fed creeks of the Ozark Plateau. I am not a university researcher. I am not a government biologist. I am a field naturalist, which means I watch things for a long time before I say anything about them. I have pulled seine nets through water so thick with carp you could not see the mesh at arm's length. I have sat on a mud bank in southern Illinois at 4 in the morning and watched an alligator gar surface in a backwater channel where the state said the species no longer existed. And I have read enough monitoring reports, enough annual assessments, enough committee findings to know the difference between data that confirms what people expected and data that makes a room go quiet. This is a story about data that made a room go quiet, and about a fish that was doing its job for 15 years before anyone in that room noticed. For years, the data only moved in one direction. Asian carp biomass in the Illinois River climbed season after season, monitoring period after monitoring period. The Illinois Natural History Survey had been tracking fish populations in that river since 1957, through what became one of the longest continuous freshwater monitoring programs in American history. The dataset it accumulated represents nearly 7 decades of population records across one of the country's most ecologically important river systems, a waterway that drains portions of Illinois, Wisconsin, and Indiana before flowing into the Mississippi. For most of that record, the data told a familiar story. Native species holding steady, then fluctuating, then declining as agricultural pressure, habitat loss, and water quality issues accumulated across the 20th century. And then, beginning in the 1990s, a new signal entered the dataset that overwhelmed everything else. Asian carp, specifically silver carp and bighead carp, both native to large river systems in China and eastern Russia, had been introduced to American aquaculture facilities in the 1970s as a biological tool for controlling algae and plankton in containment ponds. They escaped. Floods, operational accidents, and deliberate releases during the 1990s sent populations into the Mississippi River drainage. The Illinois River, which connects the Mississippi to the Chicago waterway system and ultimately to Lake Michigan, became one of the primary invasion corridors. By 2003, electroshocking surveys, a standard fisheries assessment technique in which electrical current stuns fish, temporarily allowing them to be counted and measured, were recording silver and bighead carp at 18.4 pounds per minute of sampling effort in some reaches. By 2007, carp represented an estimated 60 to 70% of total fish biomass in the most heavily invaded sections. Those numbers had one direction: up, So when the 2017 and 2018 survey reports came back showing something different in specific corridors, the researchers did not immediately trust what they were seeing. Dr. James Lamer, a fisheries biologist at the Illinois Natural History Survey, who had spent years watching population trend lines that never gave him anything encouraging, encountered the 2018 data with professional skepticism. The readings showed a statistically significant decline in catch per unit effort for bighead and silver carp in 3 monitored reaches of the lower Illinois River. His first assumption was methodological error. He ran the numbers again. The decline held. Not a crash, not an elimination, a measurable, directional, multi-season decline in exactly the metric researchers had been watching trend the wrong way for more than a decade. What made it stranger was the commercial harvest data sitting alongside it. Removal operations had not increased meaningfully in those sections. The organized commercial netting events, which had been pulling tens of thousands of pounds from the river annually, had not scaled up enough to explain what the monitoring data was recording. The Asian Carp Regional Coordinating Committee's 2019 annual report acknowledged the population softening in parts of the Illinois system, but stopped short of explaining it. The harvest numbers didn't explain it. The barrier systems didn't explain it. The data was pointing toward a cause that nobody in the report was yet naming. Something was reducing juvenile carp survival in those corridors at a rate no active management program had achieved. The monitoring data had recorded the effect cleanly. Nobody had yet identified what was producing it. Here is the detail that reframes everything that came before it. In 2019, researchers at Southern Illinois University Carbondale published findings from a multi-year gut content analysis of alligator gar collected across the Mississippi and Illinois river systems. Alligator gar, Atractosteus spatula, is the largest freshwater fish native to North America. Adults regularly exceed 6 feet in length and can exceed 300 pounds. They are among the oldest fish lineages on the continent, with fossil records extending back more than 100 million years. A species that swam through the same water as the dinosaurs and outlasted the extinction event that ended them. By the mid-20th century, that survivor had been effectively eliminated from most of its historic range, not by a natural disaster, not by a competing predator, but by the people who shared the river with it. Wildlife managers in multiple states had actively targeted alligator gar as a trash fish, a junk species believed to compete with the bass and catfish that license fees paid to protect. They used electrofishing, netting, and poisoning programs to suppress populations. By the 1970s, the species had vanished from Illinois, Indiana, Ohio, Kentucky, Tennessee, and much of the upper Mississippi drainage. The river had lost the animal that had been regulating it for 100 million years, and nobody at the time thought that mattered. Dr. Solomon David, one of the country's leading alligator gar biologists and a researcher at Southern Illinois University Carbondale, had been arguing for years that the species deserved serious reconsideration as a native ecosystem regulator. His team's gut content analysis provided the biological evidence that had been missing from that argument. What they found inside the stomachs of recovered specimens was not ambiguous. Asian carp appeared repeatedly across multiple fish, across multiple collection sites, not as an occasional dietary item, but as a consistent, documentable dietary component in gar occupying the same backwater and floodplain habitats where juvenile carp concentrate during their first and second year of life. This mattered for a specific biological reason that commercial harvest had never been able to address. Adult Asian carp are difficult prey. A mature silver carp moving at speed through open water is not something most native predators can reliably intercept. But juvenile carp in their first 12 months are an entirely different calculation. They are smaller, slower, and they concentrate in exactly the shallow, vegetated, low-current backwater habitats where alligator gar historically evolved to ambush prey. A single adult alligator gar holding motionless in cover, which is precisely how the species hunts, using its low metabolic rate and ambush strategy to minimize energy expenditure, can take juvenile carp repeatedly across an entire spawning season without expending the energy that active pursuit would require. It does not chase, it does not pursue, it waits. And when the juvenile carp drifts into range, the strike takes a fraction of a second. That is how the species has hunted for 100 million years. It did not need to adapt to the invasion. The invasion came to it. Multiply that across a recovering population of gar distributed through connected backwater systems, and the arithmetic changes. You are no longer talking about individual predation events. You are talking about a systematic, continuous reduction in juvenile carp survival rates, operating at exactly the life stage that determines whether a population grows, holds flat, or begins to contract. The math underneath this is straightforward and consequential. Asian carp reach reproductive maturity in 2 to 3 years. If enough juveniles fail to survive those first 2 years across enough consecutive seasons, the adult population stops replacing itself at the rate it needs to sustain its numbers. Commercial harvest targets adults after the damage is already done, after those fish have already fed, already reproduced, already reshaped the water column around them. Predation on juveniles targets the pipeline before it delivers. It operates on the recruitment stage, which is the only stage where population level change is structurally possible over the long term. In 2004, the Illinois Department of Natural Resources made a quiet decision that had nothing to do with Asian carp. The agency launched a native species restoration initiative targeting alligator gar. The program was framed entirely around biodiversity and ecological heritage, around restoring an animal that belonged in those river systems before humans systematically removed it through decades of misguided eradication campaigns. Nobody in that planning meeting was thinking about invasive species management. The program's success metrics were defined around survival and population establishment, not around carp suppression effects that wouldn't be detectable for more than a decade. The early stocking numbers were modest, hundreds of hatchery-raised juvenile gar released into carefully selected backwater habitats along the Illinois River and connected floodplain lakes. Hatchery production of alligator gar presents its own technical challenges. The species has specific water chemistry and temperature requirements during early development, and fingerling survival rates in the first weeks of life are sensitive enough that producing sufficient numbers for meaningful stocking events requires dedicated infrastructure. The Tishomingo National Fish Hatchery in Oklahoma, which has developed expertise in alligator gar propagation, became one of the primary production facilities supporting these regional restoration efforts. Managers estimated that a meaningful percentage of stocked juveniles would not survive to adulthood. That was expected. That was built into the program's projections. Missouri followed with its own restoration effort in 2008. Arkansas expanded its existing gar management framework around the same period. Tennessee initiated targeted stocking shortly after. Each program operated independently, reporting to different agencies with different mandates and different success metrics. None of them were coordinating with Asian carp management programs. None of them were designed to. That institutional separation, the fact that biodiversity restoration and invasive species management were being conducted as entirely parallel operations with no communication between them, is what allowed the effect to build for nearly 15 years before anyone connected the two data sets. Alligator gar are not fast-maturing fish. Hatchery juveniles stocked in 2004 and 2008 required 7 to 10 years to reach body sizes capable of consistently taking carp-sized prey. That biological timeline meant the predation pressure those fish were capable of generating did not begin accumulating in monitoring data until somewhere between 2014 and 2016, which is precisely when the Illinois Natural History Survey's long-term population trend lines, the ones Dr. Lamer had watched move in one direction for his entire career, began showing something they had never shown before. Researchers at the Columbia Environmental Research Center, part of the U.S. Geological Survey, ran a retrospective analysis in 2022, cross-referencing gar stocking records against carp population, monitoring data across overlapping river sections. The correlation between years since stocking and measurable carp decline in those specific corridors was consistent enough across independent data sets to demand a formal explanation. The Biodiversity Program had been running a carp suppression program the entire time. It just didn't know it. Here is the problem that every freshwater biologist working the Illinois River system is carrying right now. Alligator gar are long-lived. Individuals have been documented surviving beyond 95 years in wild populations, confirmed through bone and scale aging studies conducted by Texas Parks and Wildlife Department researchers. The species uses a calcified structure called the gonoid scale, an ancient scale type found in few living fish families that accumulates growth rings in a manner similar to tree rings, allowing age determination from recovered individuals. That longevity is part of what makes them effective ecosystem regulators over generational timescales. But that same biology creates a lag that is genuinely difficult to manage around. Hatchery stocking events produce juvenile fish. Those juveniles require 7 to 10 years minimum before they reach the body size capable of consistently taking carp-sized prey. That means every stocking event made today does not generate meaningful predation pressure until sometime in the early 2030s. And reproductive populations, wild gar actually breeding in sufficient numbers to sustain themselves without continued human intervention, require even longer establishment timelines because the species reaches sexual maturity at 6 years for males and 11 years for females. The electric dispersal barriers on the Chicago Sanitary and Ship Canal, the primary infrastructure separating the established carp population from open water access to Lake Michigan, were designed and built as a temporary solution. The U.S. Army Corps of Engineers' own 2022 Brandon Road Project Feasibility Report acknowledged that the existing barriers were never engineered to function as a permanent separation mechanism indefinitely. The Brandon Road Project, a proposed series of additional deterrent technologies including engineered water jets, acoustic deterrent systems, and bubble barriers, carries a projected federal cost of $858 million and has been in planning and partial funding stages since 2017. As of 2023, full construction authorization remained incomplete. Dr. Caleb Hasler, aquatic ecologist at the University of Winnipeg, who has studied fish passage and barrier effectiveness in North American river systems, published an assessment in 2022 estimating that alligator gar would need to re-establish at functionally significant densities across a minimum of several hundred miles of connected river habitat to generate suppression effects at a scale relevant to Great Lakes protection. Current recovery efforts cover a fraction of that range. The uncomfortable reality sitting underneath all of this is precise. The biological solution moves on a timeline measured in decades. The infrastructure solution moves on a timeline measured in budget cycles and congressional authorizations. And Asian carp move on a timeline measured in spawning seasons. While the debate over barriers and budgets and biological timelines continued at the federal level, something was happening at the state level that researchers had not anticipated. In 2021, Dr. Dwayne Chapman, a research fisheries biologist with the U.S. Geological Survey's Columbia Environmental Research Center, published a comparative analysis examining carp population trends across multiple river systems simultaneously. His team had been collecting data long enough to support cross-system comparisons that earlier research couldn't. The finding was specific and striking. River sections showing the steepest and most sustained declines in Asian carp recruitment were not the sections receiving the most aggressive commercial harvest pressure. They were not the sections closest to the electric barrier infrastructure. They were not the sections with the most active management intervention of any measurable kind. They were the sections with the longest continuous history of native apex predator presence. Whether through successful gar stocking, natural remnant populations, or connected habitat that had maintained some level of large predatory fish through the decades of broader population collapse. The pattern held across the Illinois system, across sections of the upper Mississippi, and in portions of the Ohio River drainage where remnant alligator gar populations had persisted in backwater habitats largely undetected by mainstream monitoring programs. One of those remnant populations was documented in Kentucky's Lower Green River system in 2019 by a team from the Kentucky Department of Fish and Wildlife Resources during a routine electroshocking survey. The crew was not looking for alligator gar. The species had not been recorded in that drainage in modern survey records. When the fish came up in the sampling gear, the lead biologist on the boat, Aaron Kindle, later told colleagues he initially misidentified it because nothing in his professional experience had prepared him to encounter one there. When his team cross-referenced carp monitoring data for that same river section, the juvenile carp recruitment numbers were measurably lower than in comparable adjacent sections without confirmed gar presence. One remnant population, undetected for years, holding in a backwater stretch nobody was watching, already suppressing recruitment in its immediate habitat range without any human management directing it to do so. The river had kept its own predator alive in a place nobody thought to look, and that predator had been doing its work in silence the entire time. What the Kentucky discovery confirmed was something the stocking program data had suggested but never cleanly isolated. Alligator gar do not need to be numerous to begin generating measurable effect. A relatively small number of large established individuals distributed across the right habitat can produce recruitment suppression that shows up in monitoring data years before anyone thinks to look for a biological explanation. The key variable is not gar abundance. It is gar establishment. Fish old enough, large enough, and habitat connected enough to be operating as functional apex predators in the specific backwater zones where juvenile carp are most concentrated. For years, skeptics raised a legitimate alternative explanation for the apparent carp declines in parts of the Illinois and Mississippi systems. Maybe the fish weren't gone. Maybe they were just somewhere else. Redistribution rather than population contraction. It was a fair question. Then the genetics came back. In 2023, researchers working under the U.S. Geological Survey's Environmental DNA Monitoring Program began comparing water column DNA concentrations from fixed Illinois River sampling stations against archived samples dating back more than a decade. Environmental DNA, or eDNA, is genetic material shed by living organisms into their surrounding water through mucus, waste products, skin cells, and scales. Unlike netting surveys, eDNA analysis does not depend on physically catching fish. It detects and quantifies genetic material present in the water column at the molecular level, providing a signal of presence and relative abundance that is independent of sampling gear efficiency and fish behavior. The multi-year comparison revealed something difficult to dismiss. Between 2015 and 2022, silver carp eDNA concentration signals declined significantly in multiple lower Illinois River sampling zones, particularly in backwater-connected stretches where alligator gar restoration had been most active. The decline was not uniform system-wide. Upstream zones still showed strong signals, but in specific corridors, the genetic load in the water column had measurably dropped. Not harder to catch carp. Fewer carp. The distinction matters. Simultaneously, eDNA began detecting increasing genetic signatures from large-bodied native predatory fish, including alligator gar, in stretches where the species had not been recorded molecularly in earlier years. The two signals were moving in opposite directions in the same geographic zones. Declining carp DNA, rising gar DNA. Dr. Wesley Daniel, a molecular ecologist collaborating with the USGS program, summarized the finding at a 2024 regional fisheries workshop. The signal shift was directional. It was not sampling noise. Something was altering biomass structure in those corridors at the molecular level. Critically, eDNA analysis also detected reduced juvenile carp genetic signatures during peak spawning runoff periods, suggesting that recruitment, not just adult distribution, was being affected. That distinction matters enormously. Recruitment suppression is the only mechanism that can produce durable, multi-year population change. Adult removal through commercial harvest reduces biomass temporarily. If recruitment continues unaffected, the population rebuilds. Only suppressing the pipeline, the rate at which juveniles survive to reproductive age, can change the structural trajectory of a population over time. The molecular data confirmed what field surveys alone could only suggest. The shift was biological, not logistical. The water itself was recording the transition. By 2024, something began appearing in Illinois River vegetation surveys that had not shown measurable recovery in nearly 2 decades. Submerged aquatic vegetation, the beds of underwater plants that form the structural foundation of healthy freshwater ecosystems, providing habitat for invertebrates, cover for juvenile native fish, and sediment stabilization, had been collapsing in the Illinois River since the carp invasion intensified in the early 2000s. Silver and bighead carp feed by filtering phytoplankton and zooplankton from the water column, a feeding behavior that simultaneously removes nutrients and disturbs bottom sediments. The result is increased turbidity, cloudier water with less light penetration that suppresses the photosynthesis submerged plants require. In 2006, average light penetration depth in heavily invaded reaches of the Illinois River fell below 20 centimeters during peak biomass years. Native aquatic plant beds declined accordingly, and with them the invertebrate communities and native fish populations that depended on that vegetated habitat. Researchers with the Long-Term Resource Monitoring Program, operated jointly by the U.S. Army Corps of Engineers and USGS, had been tracking aquatic plant coverage since the early 1990s across a network of fixed monitoring stations in the upper Mississippi River system and connected backwaters. Between 2019 and 2023, survey teams recorded incremental but statistically significant increases in submerged vegetation density in specific backwater complexes downstream of Havana, Illinois, the same regions where carp recruitment had weakened and gar presence had increased. Water clarity measurements improved in parallel. Secchi disk readings, a standardized turbidity measurement using a black and white disk lowered into the water until it disappears from view, rose by measurable margins during late summer sampling windows. Not dramatic shifts, but consistent ones, repeatable across multiple survey years, the kind of signal a system sends when something structural has changed beneath the surface. This is the beginning stage of what ecologists call a trophic cascade, a chain of ecological effects triggered by changes at one level of the food web that propagates upward and downward through the system. When juvenile carp survival declines, fewer adults reach peak biomass. With fewer large filter feeders disturbing sediment and consuming plankton, turbidity decreases incrementally. Increased light penetration allows submerged vegetation to regenerate. Plant beds stabilize sediment further and provide habitat for the invertebrate communities that form the nutritional foundation for native fish recovery. In 2023, long-term resource monitoring data showed modest increases in young-of-year bluegill and largemouth bass catch rates in those same recovering backwaters, species whose juvenile survival depends directly on the vegetated habitat that carp had suppressed for nearly 20 years. Dr. Michael Dreslick of the Illinois Natural History Survey described the situation carefully during a regional fisheries briefing. The conditions being observed were not a restoration to historical baselines, but in specific locations invasive dominance was no longer absolute. Localized biological resilience was appearing in places where it had not been measurable in over 15 years. That distinction is the whole point. Commercial harvest removes carp biomass temporarily. Predator-driven recruitment suppression alters system structure. For nearly 2 decades, Asian carp appeared to have permanently re-engineered parts of the Illinois River food web. Now, in corridors where apex predation has quietly returned through a program that was never designed to produce this outcome, the food web is responding, not because someone ordered it to, not because a committee approved it, because the river got back an animal it had lost, and that animal did what it had always done. The question the data is now forcing is not whether the biological mechanism works. The evidence from the monitoring programs, the eDNA analysis, the vegetation surveys, and the Kentucky remnant population discovery all point in the same direction. The question is whether the recovery of the predator can be accelerated enough across enough connected river habitat to matter at the scale that Great Lakes protection actually requires. Nobody planned for alligator gar to be part of the answer. Nobody coordinated the stocking programs with the carp management programs. Nobody ran the projections that would have revealed what was building in those backwater systems over 15 years of parallel, disconnected effort. The river figured it out anyway. If you want more stories where ecology produces answers nobody was looking for, subscribe and follow. Because the science developing in these river systems right now is moving faster than the policy framework designed to respond to it.